STB 1083 Biochemistry Practical 2B Determination of sugar in sample using the 3,5-dinitrosalicylcyclic acid DNS method by subgroup 8.
Hi, my name is Jolene Yoriting and my matrix number is 99321.
Hi, my name is Alexa Sherilana Jacob and my matrix number is 98785.
Hi, my name is Letitia Enlin with matrix number 98782.
My name is Ira Wahyuni binti Abdul Hadi with matrix number 99162.
The objective of this experiment are to apply the DNS method to detect and measure reducing sugar in drink sample. Next, to determine the absorbance of the sugar DNS complex at 575 nanometer by using a spectrophotometer. Lastly, to calculate the sugar concentration in sample. The material used for this experiment firstly is DNS reagent. It reacts with reducing sugar when heated to produce a reddish-brown color for measurement. Next, Rochelle salt to stabilize the final reddish-brown color to ensure an accurate reading. Cuvette, a transparent container that holds the final solution for analysis in a spectrophotometer. Hot plate provide the necessary heat to drive the chemical reaction between the sugar and DNS reagent. Glass beaker used for placing the test tube during boiling process. Distilled water acts as a solvent for creating standard solution and used as a plain reference. Test tube and rod serve as vessel for the reaction and are held by rack for organization and safety. Micropipette and tips used to accurately measure and transfer precise small volume of liquid. Selected drink sample, the substance being analyzed to determine its sugar content. Firstly, a total of 12 clean and dry test tubes were prepared and labeled as 1A, 1B, 2A, 2B until 6A, 6B, with each condition prepared in duplicate. The volumes of sample solution and distilled water added to each test tube were as follows. Figure 1. Next, 1 milliliter of DNS reagent was added to each test tube. All test tubes were placed in a boiling water bath and heated for 5 to 7 minutes to allow for color development. The test tube was cooled down at room temperature after boiling. 1 milliliter of Rocher's hard solution was added to each test tube to stabilize the color formed during the reaction. The solution in each test tube was transferred into cuvette for analysis under spectrophotometer. The color development of the mixture was read at 575 nanometer by using a spectrophotometer. Test tube 1 was the blank set as negative control to eliminate background readings. In conclusion, the experiment was a success in all three objectives. Firstly, the DNS method was used successfully in identifying reducing sugar in the drink sample. There were different absorbance values, with test tube 1A and 1B having the highest, while test tube 6A and 6B did not record any absorbance. This means that the amount of reducing sugar reduced from tube 1 to tube 6. The absorbance of the sugar DNS complex was also properly taken at 575 nanometer through the use of spectrophotometer. Furthermore, the absorbance result can be used with a glucose calibration curve to estimate the amount of sugar in each sample. The higher the absorbance, the greater the content of the sugar. Group reflection, new learning. We learned that the DNS method detects reducing sugar by converting them into a colored compound, which can be measured to estimate sugar concentration. Next, we learn how to analyze absorbance data, apply dilution factor, and use a standard curve to determine the glucose concentration in the samples. Challenge phase. The experiment encountered difficulties with proper sample dilution preparation and precise pipetting, especially when working with small volume. To guarantee accurate results, the glucose concentration in each dilute sample had to be within the spectrophotometer measurable range. Extreme dilution may produce low absorbance, while high concentration may produce inconsistent results. To obtain the ideal concentration for precise reading, careful planning and computation were required. Suggestion for improvement. The experiment suggests the use of easy-handling, beginner-friendly pipette such as digital or adjustable micropipette with ergonomic grip and soft plungers to improve accuracy and comfort during the experiment. This would eliminate the stiff plungers and inconsistent sample measurements.